Hello, my name is Ziqi, and I'm a PhD student in Georgia Institute of Technology. Today, I wish to introduce to you our recent work, scDisInFact, disentangled learning for integration and prediction of multi-batch, multi-condition single-cell RNA-seq dataset. In disease study, single-cell RNA-seq are measured from samples of patients under different experimental conditions. And such condition can be the demographical information of the patients, disease severity of the patients, or drug treatment of the patients. And the biological differences exist between samples under different condition groups. There are also technical batch effects that exist between samples of different experimental batches. And we term such datasets as multi-batch, multi-condition datasets. And our model, scDisInFact, mainly deal with such multi-batch, multi-condition dataset, and it performs 3 tasks jointly, including the removal of batch effect, the discovery of condition-associated key genes, and the condition effect prediction, or we call it perturbation prediction. scDisInFact is designed based on a variational autoencoder framework. It disentangled the latent embedding of the VAE model into the shared biological factors here, and a bunch of unshared biological factors and batch factors. And shared biological factor encodes the biological variation that is irrelevant to the condition effect, which can be the cell type variations. And the unshared biological factor mainly encodes the condition effect for different condition types, which can be the drug treatment, disease severity, or the age group of the patients. And there is also a batch factor, which is like one hot batch ID that encode mainly the batch effect. And this is the framework of scDisInFact model. The latent embedding are fully disentangled, and the shared biological factor can be used for cell type identification and cell clustering, as it is removal of both condition effect and batch effect. And the first layer of the unshared encoders can be used to extract the condition-associated key genes. By updating the unshared biological factors and batch factors, we are able to make perturbation prediction. It's to predict the gene expression data of the cells under different conditions. We test our model scDisInFact on simulated datasets, where we measure the latent space disentanglement, key gene detection accuracy, running time, and perturbation accuracy of our methods and baseline methods. And the comparison result shows that our method consistently perform better than baseline method. In the end, we test our model on a real dataset. It is called a glioblastoma dataset. And the dataset includes 21 matrices from 6 patient batches, with and without drug treatment. The drug is correspond to the panobinostat. And we feed the dataset into the scDisInFact model and visualize the learned shared biological factor and unshared biological factors using UMAP. The plots here shows the visualization results of shared biological factor and unshared biological factors. We can see that shared biological factors separate the cell of different cell types and at the same time remove the condition and batch effect. On the other hand, unshared biological factors separate the cells of different conditions and remove the batch effect. In addition to that, we extract the top scoring genes learned from this dataset, and we found that these genes are highly correlated to the neuron markers and the macrophage markers, which were reported to be relevant to the drug treatment. The top gene ontology terms also related to the drug treatment. And this shows that we are learning the correct condition-associated key genes. In the end, we test the perturbation prediction accuracy of our model, and we measure the alignment of the predicted distribution and the ground truth distribution using several metrics, and compare scDisInFact with baseline methods. And comparison results shows that our method consistently perform better than baseline method. And we have our software available on GitHub and paper available on bioRxiv.